Welcome to Decoded, the podcast that explores how data, technology, and human insights are reshaping patient care worldwide. I'm Jurgi, Camblong, biologist, dreamer, and founder at SOPHiA Genetics. In each episode, we decode insights with the pioneers using data not just to imagine, but to create a new reality in healthcare. Today, we're taking a closer look at cancer, what it is, how our understanding of it has evolved, and how new tools from targeted therapies to liquid biopsies are reshaping care. My guest is Pedram Razavi, who leads breast oncology and precision medicine programs at Memorial Sloan Kettering Cancer Center in New York. His work in genomics and liquid biopsy is helping us see cancer in ways we never could before, from tracking how tumors evolve in real time to detecting disease when it's almost invisible. Together, we will explore the history, the science, and the human side of oncology and what the future might look like. Let's dive in. Hi, Pedram.
Hi, Jurgi. Thanks so much for having me. Excited to be here.
So, Pedram, to start with, you're the first oncologist I'm interviewing for my podcast. And I thought it would be interesting if you could tell us, when was cancer described for the first time? And actually, while I'm asking you that question, this might bring you back to your med school eventually.
The first mentions of cancer and tumor actually was not called cancer at the time, goes all the way to the ancient Egypt. It was mentioned, and then later on in the Rome, there was also, and Hippocrates started talking about carcinos and cancer based on the way it was grown, and then the Romans expanded on that. And then during the golden era of Islamic civilization in Iran, Persia, there were two scientists and philosophers, physicians also, Razi and Avicenna. I got the book of Avicenna as a gift when I graduated med school. And actually, I read some parts of it. It's very, very exciting how they describe different types of cancer. There was a lot of surgical stuff in it, as far as I understood at the time, but it was surgery and there was a lot of emphasis on clean margins, something that now the surgeons are, obsessed with it. They were talking about getting the clean margins extracted, otherwise it's going to recur. And there was also some herbal medications, some of them later proven to have anti-neoplastic effects. But, I think the major inflection was when the microscope was invented and Virchow started looking at the cells, and others have identified cancer cells, that they are, how different they are. And then we started to have, different better treatments, chemotherapies, radiation therapies, better surgical techniques. Anesthesia came on to help with the treatment of cancer. And then in the past few decades, there's been a huge evolution and revolution in understanding of cancer, understanding molecular aspects of the cancer, genomic aspects, different treatments, so on and so forth. So it's been quite a journey.
Perfect. Thank you. And actually pretty fascinating listening to you that it started so long ago, right? Because obviously we know that the environment influences as well our body, and maybe, can be sometimes cause of cancer. But some people believe that sometimes cancer is new, but as you describe it, is kind of intrinsic to our biology.
Yeah.
And so along those lines, so thanks for taking us back to thousands of years and then the microscope and now the days of molecular biology. Can you explain us what is today our understanding of cancer? How do we define it? And how this definition has eventually evolved over the last decades?
So, our understanding of cancer has very much evolved. But I think if you think about this, these are a group of cells that decide to be unruly and not follow the principles that every other cell in the body abide to. So they proliferate. When there is a signal to die, when there is something wrong with the cell, there is always a signal to die. We call it apoptosis. They evade that. They find a way to invade other tissue and the natural boundaries that are sacred boundaries in the cell. The cells usually don't pass those boundaries. These cells learn how to do this, increase their blood supply, increase their vasculature, increase their metabolism, change their metabolism, get more energy disproportionately. So these are bunch of, cells that, selfish, they want to invade, they want to grow, similar to virus and other types of infections that they come to body and then use the host. These are our own cells that somehow they try to be unruly. And as an oncologist, when we look at cancer, and as I started to gain more experience in being an oncologist, we get a sense of what cancer is. There is a feeling, you see a tumor, and after a few times that I treat this patient, I get a sense what type of cancer I'm dealing with. Is this one of those tricky ones or is it not? And then a lot of it nowadays goes into the biology of it, and some of it we can explain, thankfully.
And we'll get there. We get there to the intimacy with cancer and
Yeah.
at some point, and the dumb versus the smart cancers. But before we get there, so thanks first for this explanation. And actually remind me a book from Richard Dawkins, right, on the selfish gene. And what you describe is cancer is kind of selfish and needs to play with biology and his own strengths to be able to escape death. I heard as well recently, Pedram, from an oncologist describing cancer on a similar way than you did, and then telling me, but from my friend in the field of immunology, for her, the cancer is rather our body was not being able to defend against cancer, right? And
Yeah. That was just recently when Hanahan and Weinberg updated the hallmarks of cancer, and they updated it now 3 times. That was added as one of the hallmarks, how to evade the immune system. And also now we are learning more interestingly that they also actually use the signal that comes from immune cells synergistically to help them grow. So actually they sometimes employ the immune cells to help them evade and grow as well. So it becomes a very, very complex issue. Yeah.
Incredible.
Fascinating.
Fascinating indeed. So speaking about fascination, you grew up, as you said, in Iran, where medicine has been practiced, much before in Western countries, definitively. What did motivate you to work in oncology?
Yeah, so I grew up in a family of engineers, Jurgi. So everyone in the family are engineers, scientists, Yeah, yeah, I think it was because it wasn't for my mom, I wouldn't go to medicine. She somehow implied, and she knew me well, if she pushed me hard to something, I wouldn't do it. But now, in retrospect, I think she really wanted me to be a doctor. So she just suggested in high school that, you've been exposed to a lot of engineering stuff and you're good in math and physics and whatever. Why don't you go and do volunteer in the hospital? So I started volunteering in the hospital, and that was the beginning of it. So the fascination started there. And then I knew that I'm going to become a doctor. And in Iran, you decide to go to med school after high school. So it's the combined bachelor and med school program. So when I was 17, I went to med school. The oncology part also came pretty early. So I was 19 or so. My dad was diagnosed with glioblastoma. As you know, it's one of the worst types of cancer anyone can get. And it was very obvious that it was a lost battle from the very beginning. When he had it, he was declining very quickly, so on and so forth. But that prompted me to hit the library. So I spent a lot of time in the library, and it was an era when oncogenes were being developed, discovered, tumor suppressors are being discovered, the new treatments were coming, new technologies, So at some point, I was not even reading about glioblastoma. In the beginning, I went to read about glioblastoma, and then I started reading about all sorts of cancer biology, so on and so forth. And then after I finished med school, I came to US. I wanted to do genetics. I wanted to do cancer. So I joined USC, which used to have and still has a very good program in cancer epidemiology and population genetics. So I did my PhD with them, learned a lot about pre-search methodology, population genetics, and gene interactions, gene environment interaction, those type of things. Then I did a postdoc, did my residency back in California, postdoc in Boston, residency in California. I came back and I started my fellowship at MSK, and I wanted to do cancer genetics. I wanted to focus on germline. That was my background. And my late mentor, Jose Baselga, just came to MSK also at the same time as I did. And he gave a grand round on one of his work, from the lab that later became a Nature paper on parallel evolution and convergent evolution of breast cancer under therapeutic pressure, resulting in resistance to PI3K inhibitor, which was a very new treatment at the time. And I was sitting there as a first-year fellow looking at this and said, this is a lot of the stuff that I learned in population genetics, cancer epidemiology can be used in the single patient. And then I immediately emailed Jose I said, okay, I want to do this. And he said, come over. We had a meeting, and then I joined his lab the following year in my second year and got a lot of mentorship, switched my field from cancer genetics, pure germline to and cancer susceptibility to cancer genomics. And then the liquid biopsy came. There was a whole evolution and revolution, genomic evolution at the time. So it was a very interesting timing when I joined oncology. And I would say that one event completely changed my future. That one talk inspired me to switch my field. Although I should say the germline and somatic borders are not as clear as they used to be. Now we know that the somatic alterations in the tumor also are very much affected by the germline alterations as well. So now we are doing a lot of that also in parallel to what we've been doing.
Great. So thanks first for sharing your personal experience, your motivations. And amazing to hear as well. It's very inspirational how, while you were equipped with some knowledge, experience, you're being able to translate that into oncology. And so moving, as you said, right, from germline, so inherited cancers to acquired cancers, you've seen precision medicine evolve during your training. And so are we thinking about oncology the same way today versus 10 years ago? Or what has been the biggest shifts?
Fascinating question, Jurgi. So I'm going to give you an example about breast cancer. And a lot of precision oncology actually started in breast cancer, interestingly. So it goes back to late 1800s when Thomas Nunn suggested the connection between the ovaries and the advanced breast cancer, and Thomas Whitson did the first bilateral oophorectomy to remove the signal from ovaries, estrogen, and then documented that these advanced breast cancers, some of them progress and respond to that type of treatment. And then obviously later on, estrogen receptors were discovered, anti-estrogens came, then anti-HER2 therapies came, and completely evolved the way that we practice breast oncology. But even in the past 10 years, a lot has changed. Or in the past 15 years. When I started, it was the beginning of clinical genomics. So there was the time that TCGA papers were being published left and right. It was easier for us to sequence the tumor, sequence the whole genome. Clinical sequencing became a thing. So we started sequencing every patient.
And this was
around 2012, 2013.
Yeah. So not so long ago,
not
right?
so long ago.
Yeah.
Not so long ago. That's when we started using them. So maybe around a little bit earlier, we started sequencing, but 2012, 2013, when we started using it very routinely in clinic. And then we thought that we have solved the problem. We're going to sequence everyone and we're going to find the major drivers of the cancer, and each cancer has a few drivers. We target them and these cancers start responding. But in reality, not every cancer was as easy to treat as we expected. So there was a huge amount of interaction between the genes, huge amount of plasticity in breast cancer. And the tumors and patients responded to treatment for a while and then immediately started developing progressions and developed mechanisms of resistance. And some of the tumors that, had all the targets and we expected them to respond, they didn't respond at all to the treatments, to the appropriate treatments. So that was the beginning of how the precision oncology evolved. So we started monitoring the patients routinely during the therapy. We started using liquid biopsies. We started doing pre-treatment, post-treatment biopsies, comparing the pre and post-treatment biopsy. What has evolved? Why this tumor didn't respond? How this tumor changed its genome or epigenome to become resistant to certain therapies. Then there was a lot of, I would say MSK is fantastic in doing this huge amount of interaction between clinicians, translational scientists, and basic labs and translational labs. So we started having xenografts and cell lines made from our patients, studied them at the same time. So when the patient goes on a treatment, a xenograft, a cell line made of that, in parallel, that tumor is being studied in the lab. And when we see the clinical findings, we try to understand them based on the similar models in the lab as well. And then some of our patients at Mirabel, they participated in our autopsy programs. It's impossible to get samples from every single site. You just biopsy one site, that's it. So there's a huge amount of heterogeneity in cancer. So we started also using this multi-regional samples from autopsy to understand better how these tumors evolve to become these lethal tumors. What's the characteristics of that lethal clone? And started putting this together. And this whole program is helping going back and find who is responding, who is not responding, how better we can select the population of patients that would or would not respond to certain therapy. What other combinations from the lab is suggested to help us overcome the cancer, and then put together these type of characteristics to design the next generation of trials, so on and so forth. So this has been a huge evolution. The way that we are doing it, we do not look at breast cancer the way that we've been looking at breast cancer 10 years ago. 10 years ago, we only had ER, PR, and HER2. And now more than 60 to 70% of estrogen receptor positive breast cancers have an FDA approved targeted therapy. That it's just that list is just expanding. That list is going to increase daily the way that we are expanding our horizon here.
Great. Thank you so much. And so basically, at some point, we thought with the sequencing and the identification of mutations that we had finally cracked the code, and then realized it was more nuanced than that. And as you explained, this requires still a lot of experiment, even on tissue biology, cells, and so on, to experiment new treatments and understand, I think, better what I love you call the cancer history, or how you define it already, the natural history of cancer.
Yeah.
Can you tell us a little bit more of the natural history of cancer and its plasticity, and why all these experiments are important to see eventually how similar cancers might evolve over time, and how this is eventually helping us better anticipate how to use the arsenal of treatments, right, as the cancer is evolving?
Yeah, so far we've been playing catch up, Jurgi, with cancer. So we give a treatment, the cancer evolves, changes, we see how things go, we test them, we get a biopsy, we get a liquid biopsy, we look at the tumor DNA in the blood, and we find a new mutation. And then we try to go after that particular mutation or particular target or mechanism of resistance, so on and so forth. And what we've learned that most of the cancers are not dumb, or as dumb as we hope they would be. There are some cancers that, you give them treatment and they disappear. And, those are the success stories. And those are the ones that we love to see more and more in clinic. But majority of the cancers change and they have a lot of capabilities. But what we are learning, and I think that's a new fascination in our group here, is that these are not omnipotent cancers. They cannot go and develop every single mechanism of resistance. If there are 20 different mechanisms of resistance to this particular therapy, this cancer can potentially develop 1, 2, maybe 3 of them, not every single one of them. And in majority of the times, they actually like one of them better than the other one. And it's because of the way that the genome is, the way that the germline background of the tumor. Some of them are just potentially lethal to that tumor. Some of this new mechanism of resistance are just against their survival. So they have some restrictions. Now we started to learn that we can actually potentially predict the mechanism of resistance before it happens. That for the first time put us ahead of the cancer. So we can be proactive. And now we have some examples in our group and in our lab. The paper is under revision that, copy number profile of the tumor and the number of copies of the genes that the tumor has can dictate what type of mechanism of resistance this tumor is most likely capable of. Then the tumor has the different mechanisms to have genomic instability. We call them mutational signatures, so on and so forth. That also combined by the copy number and the number of these genes can affect the mechanism. Now we are developing a whole program, try to predict the mechanism of resistance before it happens. And learning from our autopsy program, it's fascinating that how this parallel convergent evolution works. Majority of these tumors go after 1 or 2 of this mechanism of resistance. So if we know that in advance and put a huge block in front of that tumor, this tumor or majority of these cancer cells may not survive. So I think that's a whole new thing that we would like to push to be more proactive and don't let the cancer develop the mechanism of resistance. Try to get the most out of our best treatments. And it's a good time in oncology. We have a lot of new drugs. We have a lot of new treatments and new combinations. So it's not, when I started, we had to rational treatments. There was like 4 targeted therapies in breast cancer. I don't want to give you this. I want to get the most mileage out of this one and give you the next one, so on and so forth. Those days are gone. Now we have too much. We have too many treatments. How can we use them better? How can we use them to eradicate cancer, potentially even in metastatic disease? And also, now we have a lot of technologies to monitor cancer as well on treatment with liquid biopsy, so on and so forth. How can we use that to inform our treatments the same way as well?
Great, great. I love it. So while I was listening to you, I was imagining myself in your shoes, playing poker against cancer, but seeing the hand of cancer, right? So that you will know how to anticipate the next moves and how to beat cancer.
Yeah,
Super inspirational.
At least most of the cards, not all the cards,
most
but
of
at
the
least
cards. No.
most of the cards. And if you have a way to know it, then there is a way for us to move forward. And I have an example, I would say there's been a huge development in just by the invent of internet and the way that they track us. Human behavior is very complex. But as you can see, they have cookies on every one of our browsers. We know exactly where we go. They know the location of us based on where is our cell phone, what I buy, what I do, what websites I visit. So they have too much information about us. And now they can predict our behavior actually pretty well. It's very complex human behavior. I go and ads are becoming more targeted, more to the point every day. Why can't we do the same thing with cancer? Why can't we get a lot more information from cancer, try to predict this behavior before and try to intercept it? And I can see that happening now. It's now, the data is there. AI is coming to help us for sure. We have now the sequencing technologies are becoming cheaper and cheaper, better technologies for sequencing tumors, and also molecularly profiling tumor is coming. So I think there is a whole potential for us to put cookie on every one of those cancer cells and try to predict them.
Absolutely. And, me being provocative on the engineers, obviously we need them, right? And we need them in our battle in healthcare, where there is a lot to do, as you pointed out, on technology and AI and so on. Along those lines, you maybe already have answered to it to some extent, but what are the technologies that you're seeing revolutionizing our understanding of cancer, on our way of treating patients in the next 5 years?
From the diagnostic translational standpoint, and as you know, I'm very much involved in liquid biopsy and I'm very passionate about this. I was lucky when liquid biopsies started. That was the time that I was starting my training.
Can you take a bit of time for, people who are even not biologists to understand what liquid biopsy is about? Because I think it's a very important concept that, most of the people don't understand.
Yeah. So liquid biopsy is essentially a blood test, and it is looking at the tumor DNA or RNA from a blood test. And what we've learned, and this is not new, it goes back to 1940s when electron microscopy was invented and they started seeing fragments of DNA, Mate and Mendel, in every single one of the fluids. So they started looking at plasma, then, urine, everything. They had a whole series of papers looking at it. So this is not a new concept, but in the past few years, we've been able to characterize tumor by looking at the fragments of the DNA in the blood. Most of the fragments are coming from the normal cells, but tumors also shed their DNA in the bloodstream. And now we have a very good technology to look at the tumor DNA, distinguish it from normal DNA. And we've started characterizing tumor based on this DNA. So these fragments of DNA keep the characteristics of the cells that they are shed from. So we started using those characteristics to understand the biology of the tumor. It potentially can give us a more holistic view of the tumor genome and the landscape of the heterogeneity of cancer. The tumors are extremely heterogeneous. No two cancers, at least in the solid tumors, that are exactly the same. So it helps us understand the heterogeneity of the cancer, monitor the heterogeneity of the cancer and evolution of a cancer, because, serial blood draws is not a big deal. Serial biopsies is impossible to do. And then also now we have pushed the limits of these type of technologies to identify evidence of cancer in the blood, even at extremely, extremely low levels, what we call minimal residual disease or MRD. And that is when we go after, is there any evidence of cancer in the blood? And these are very sensitive and specific tests. Now, tests can go and find if 1 out of 1,000,000 fragments in the blood are coming from the tumor. The test, very confidently can call them and say, oh, this is cancer. But I think what excites me is not the technology part of it. What excites me is how we start using them in clinic, how we can understand about this cancer, monitor this cancer, and try to eradicate this cancer as the best that we can. And we have started looking into this. So now we can characterize the tumor based on the baseline liquid biopsy and also the tumor molecular profiling. There is now whole technologies on spatial multiomics, tumor DNA sequencing, so on and so forth. But then also monitor the response. Is the tumor DNA going down? Is the tumor DNA going up? And then adapt our treatments based on this. So very recently, a phase 3 clinical trial was reported actually showing proof of concept. It's not the best design, but it was a pivotal study, in my opinion, showing that change of therapy based on just the ctDNA changes can improve the outcomes of our patients. I think that's just the very beginning. We can now have very adaptive treatments, change the treatment based on the ctDNA response very early. That helps us not to impose a lot of side effects and toxicity to our patients for a treatment that is not effective. At the same time, try to identify combinations and therapies very early on when the tumor has less heterogeneity, potentially more susceptible to our treatments, try to eradicate it. And I'm excited. We have a clinical trial. I can probably announce it here, called Horizon Breast. It's a curative intent clinical trial in metastatic breast cancer. Metastatic breast cancer is considered to be not curable. But now we feel comfortable. We have a lot of good treatments. We have a lot of new technologies to monitor disease. We now have designed, exactly based on these paradigms that I explained, a clinical trial try to go after a subtype of breast cancer called HER2 positive breast cancer, try to eradicate metastatic breast cancer, cure the uncurable. And I think this is just the very beginning. We understand what groups of patients would or would not respond to certain therapies. We give them the best treatment that we can offer them. We monitor them, but we just modify our treatment very early on, try to go for the combinations and novel therapies that can eradicate the disease.
So basically another tool, still, I would say, not very well understood by patients and even by some doctors. So-called liquid biopsy, all of you follow that technology that further strengthen your ability as you play poker against cancer to be able to see more of the cards of cancer.
Yeah. And I think, Jurgi, I think what is going to be extremely, extremely helpful here is going to be AI. And we are
combining the data,
combining this data, because, it was impossible for us to analyze this much of it data and also make sense of and also these new technologies, new ways that we are looking at AI. Doesn't need huge amount of number of patients, even with small number of patients, but huge amount of data can learn. The whole revolution on developing foundation models. Now there are multiple oncology foundation models that learn from every single one of these patients. Then they can decipher this number of even smaller events better than before. So I think putting that also combined with all the other technologies that we have to characterize the tumor, it will help us predict the behaviors of cancer better, monitor its behavior better, match the best treatment to the patient, and also be adaptive. Try to improve our outcome of our patients.
Great. So thank you so much, Pedram. So we covered well, I think, aspects which are very important around technology, science, treatments. Now, I would like to go back to the doctor, to the medical doctor, okay, who I think you kind of gave already some indications with the intimacy around cancers, and this is part of the art. So beyond the technology, medication, how are you using your experience and sense to support patients?
Yeah. And that's probably one of the reasons that I'm staying in academia. Honestly, it's the passion for patient care. As doctors, when we practice, I think there is some epigenetic changes in our DNA and it stays with us. So I really, really enjoy clinic and the patient care. I get a lot of inspiration from my clinic. But and we are, obviously the patients come, especially for the first times when they come to see me, they come with the stress of being diagnosed with cancer. And I'm seeing a human being for the first time. They lived, 50, 60 years of their lives. And I have no idea how I'm talking to. So when I see my patients, and I've already reviewed all the records, I know exactly what's going on, everything. But I ask them to tell me about their cancer. I give them time to go over their history. And the main purpose, honestly, is to learn about who am I talking to? What type of person? What's their understanding? What's their baseline understanding of the disease? And then at the end of the day, our goal as oncologists is to make partners from our patients and make joint decisions. And the better they're informed, the better they understand their disease, and the better I understand them, I can give them better recommendations. I think that those are the parts, and that human aspect of oncology is something that can be lost rather quickly now that we are doing a lot of guidelines. Yes, you come here, the guideline says treatment A, treatment B. Soon AI is going to tell us what to do. So AI is going to tell us, oh, I have all the guidelines, I know all of this, so this is the best treatment. But at the end of the day, we have a person in front of us, and that in human interaction is incredible and should be part of our practice. And that's what I cherish in my daily and weekly. I have 1 day clinic a week. So in my clinic day, that's what I look forward to. Also, I should say as a researcher and scientist, it gives me a lot of inspiration. The failures in clinic, this patient should have responded to this particular treatment and didn't. That bothers me. That gives me a lot of inspiration to go back, understand, look deep into this case, what happened, why it didn't respond, and also the success. And I should say a lot of our work coming out of our group is actually inspired by one patient did or did not respond to this. That started the whole program. And we have a molecular tumor board at MSK that, many of us sit together and review the cases. I would say those molecular tumor board cases are a huge source of inspiration for the whole translational group here at MSK, and is helping us tailor our treatments better, tailor our approaches, not just for research, but also for the next generation clinical trials to the needs in clinic.
Great. And yeah, in the past, I had said often, I think it goes along the line of what you described, that eventually AI can help us manage some of the complexity, which should kind of intend to humanize medicine, because it should give more time to the oncologist, to the doctor, to do exactly what you describe and use our senses, our emotional intelligence to understand the patients.
Interesting that you brought this up. And this is happening more and more in clinic.
Right.
My patients, now they all have access to the charts and they can download the charts and get them, so on and so forth. One of my patients, she was a very complex case. I've been treating her for quite amount of time, few years, and she was extremely sick in the beginning. So we had to do a lot of maneuvering, different types of treatment, try to control this cancer. And we've been successful. So just very recently, she came and said, "Oh, Dr. Razavi, thank you so much for being my doctor and taking such a good care of me." And I said, "Oh, thank you. But how come, I've been taking care of you for many years?" Said, "Oh, I put all of my records in ChatGPT and ask very simply, is my doctor treating me well?" And then, ChatGPT actually very nicely said, "Oh yeah, your doctor, you have metastatic disease." It was some disclaimers, obviously, and then started saying, "That seems like you've been treated according to standards. You've been treated well, so on and so forth." But I actually saw the output. It's actually very helpful for the patients. They put their PET scan results, they put the other results into it. And at the end of the day, my time with the patients and the staff time with the patients are limited. So AI can help answer some of their obvious questions. There is a danger that, they might get information that might not be relevant or don't understand the question. So there's a huge risk with it as well. But if it's done well and in a controlled environment, can actually help us be doctors again explain things to the patients, try to answer their questions, but at the same time, they also help them understand this medical jargon and their results better in a form and a language that they understand and they comprehend. I agree with you. I think there is a very good potential that AI can help us humanize medicine and cancer care. And the other side of it is dehumanize, and just tell us what to do. And that's the danger.
Agree. So maybe to close our fascinating podcast today, Pedram, would you have some last words for the next generation of doctors and eventually next generation of oncologists?
Yeah, I think I just said it. I think it's what's important is to keep the connection and inspiration from clinic. I think it is, to me, as a translational, especially for the translational scientists. So some scientists are basic scientists, so on and so forth, things like that. But for the oncologists and the researchers who are practicing as well, I think it's important to make sure that we are not just following the guidelines and we are not just following, what we are told to do, and try to be doctors and try to solve every case. Every case, every cancer is complex. No two cancers are the same. Try to understand it. Try to understand the complex matrix of that patient. And if every time we treat a patient, this matrix is becoming more and more complex. One site responds, one site doesn't respond. This one doesn't respond. The tumor genome evolves, the biomarkers evolve, so on and so forth. Try to understand it, put it in the context, and use all the technologies that we have to help us make better decisions. But I think keeping the connection with clinic and inspirations from clinic, I think that would be my only advice.
Pedram, thank you for taking us in your journey today. From the first descriptions of cancer to how precision medicine and liquid biopsies are changing the way we detect, treat, and even think about this disease. This was really fascinating. If this conversation gave you new ideas or hope, subscribe to Decoded, leave a quick review, and check out our other episodes. And if someone in your life has been touched by cancer, share this podcast with them. It may help them see what's possible today and what's coming next. See you next time.